In this video, we're going to take a look at how you can use something called a test cross to determine the type of organism and the type of genes that they have there. So if we take a look at our monohybrid cross results from before, this is something that Mendel did back in the mid-1800s again. You take your two true breeding parents, in this case ones with purple flowers and ones with white flowers, and remember, true breeding means they consistently have that same pattern coming out, and when you cross them, the first generation had those purple hybrid flowers, and when you cross those ones with each other, you come out with that really kind of strange result that confused Mendel at first, that you have some purple flowers and you have some white flowers. And if you remember, this is how he came up with that idea that the purple trait overshadowed that white flower trait. So we have our dominant and our recessive. And when we look at this, we see the type of flower here is purple. That's our phenotype. And we also have our white flowers. So there, you're looking at what is actually expressed, or what trait or characteristic comes out from the genes that are passed on from the generations. And if you also remember, we have our genotype. Some of those flowers in that first generation have homozygous dominant, both alleles there. So in this case, we use that capital letter P for both of these genes on the chromosomes to indicate that it is homozygous. Both of those chromosomes have that same gene in the zygote that came when that first cell was produced, when the two cells fused, the two haploid cells fused together. We also have some heterozygous, that's our F1 generation, and also some of those are found in the F2. Those are the ones where they have different forms of those genes in that single zygote cell. And we also have our homozygous recessive cells, where both copies that they inherited are the recessive copy. So if we look at this, how can you determine the genotype of an organism that has the dominant trait? And think about our phenotype for the purple flowers. Well, what's their genotype? Do they have homozygous dominant, or are they heterozygous? We can't tell just from looking at them by that physical appearance there. And if you think about this process here, those homozygous dominant plants that are out there, they have those two alleles. And so when that cell goes through meiosis, you're going to produce these gametes. Both of these gametes have the same type or trait of the gene there. When you look at your heterozygous ones there, one of those copies is going to give you that purple trait, but then we have this last little one over here. This one, if it's passed on to the next generation, is going to give you that recessive trait. So how can you distinguish between those types of cells that are out there? Well, if you have a homozygous dominant purple plant that's out, or purple flowers on the plant, the possible genotype here is those two capital P letters right there. And what we can do is do a test cross with a recessive plant. We know what the genotype is for the recessive because by definition they have to have a homozygous recessive in order for that recessive trait to be observed as the phenotype there. So if we cross these guys with each other, drop it into your Punnett square. You go through meiosis and you separate those alleles, and there is one allele on that gamete and the other allele on that gamete. And likewise for our white plant over here that has the two recessive alleles. And if we drop this in, notice what happens with the progeny right there. All of the progeny that come out of this process are going to have the dominant phenotype because you have that homozygous dominant right there in the parent plant. Now think about what's going to happen if you were looking at that for the opposite case, where you have a heterozygous purple flower out there. You separate those two alleles and look what happens. Now that heterozygous allele is going to get combined with our recessives, and notice the difference for what we see in the progeny right here. So if we line these guys up and think about this unknown, if it's a heterozygous and we do that test cross with the recessive, how are we going to see the difference here? If you have that dominant trait, all of the progeny that come out of there are going to be dominant. But if your dominant is the heterozygous, then you're going to start to see this 50-50 mix in the progeny. So by doing a test cross with a recessive organism, in this case our homozygous recessive was white flowers, we can actually use this process to determine the genotype of the previous generation. Granted, this does require you to look at the progeny from that process, so you don't determine the genotype immediately. You do have to wait for the phenotype to come out in that next generation, but at least using this process, you can determine the genotype of something where they have the dominant phenotype.